Hi, I'm Dr. Charles Raison. Today we'll be talking about a study recently published in The Lancet. The study is titled Identification of Risk Loci with Shared Effects on 5 Major Psychiatric Disorders, a Genome-Wide Analysis. This is a very large study, in many ways a very striking and novel study, and it's gotten a great deal of attention around the world, both from scientists and clinicians. The primary finding of this study is, in fact, that if you look for genetic predictors or genes that are associated with psychiatric conditions, that if you look at 5 major disorders, the disorders for which they had genomic data in this study, and that was autism, attention deficit hyperactivity disorder, major depression, bipolar disorder, and schizophrenia, rather than finding different risk genes for each of these disorders, what they found was, in fact, that these disorders, although they seem to be so different from each other, actually genetically seem to be very common. To say it another way, the same genes seem to increase the risk of getting any one of these disorders. The effect was especially strong for the disorders that happen in adulthood: major depression, bipolar disorder, and schizophrenia. So using GWAS, this advanced technology, what the investigators found was that, in fact, rather than having different genetic risk factors, it appears that these 5 widely disparate disorders, ADHD, autism, major depression, bipolar disorder, and schizophrenia, in fact shared genetic risk factors in common. Now, this, of course, is biologically very important, but it also says something that is important in critiquing our current diagnostic system, which is we tend to think of these things as being discrete disease states. The genome seems to be telling us, in fact, that they're connected in deep and perhaps mysterious ways, clearly ways that we do not fully understand yet. So if we look across all the studies that have been done of psychiatric genetics, and there are very many of them, this is one of the most important ones done to date. It's got one of the largest populations. It's very well done. Now, it has certain weaknesses. So for example, they only looked at what are called SNPs, single nucleotide polymorphisms, using a GWAS technology. This allows you to find certain types of genetic risk factors, but they don't see other types that have become increasingly more relevant. So for instance, there's a thing called copy number variants, which are changes in whole large parts of the genetic material. These things often arise spontaneously. They're very rare, but they can have very large effect sizes. Those sorts of potentially very relevant genetic predictors cannot be identified by the type of methodology that they used in this study. What this study does show is that certain very common genetic alterations in genes can impact these disorders, and the same variations seem to impact a wide range of conditions. The other thing they found was that one of the genes identified is a thing called CACNAC1, which is a calcium channel gene. That gene actually codes for the alpha-2 subunit of the L-type voltage-gated calcium channel, kind of a mouthful. Why that gene is interesting is that it has been repeatedly implicated in bipolar disorder and depression, and somewhat in schizophrenia, by other methodologies, and it's known to have impacts on brain functioning that are very relevant to mood disorders. So the fact that they discovered that using this technique strongly suggests that calcium channels, L-type voltage-gated calcium channels, are far more important to psychiatry than we currently appreciate. So, you know, the DSM-5 underwent a great deal of haggling in its production because there was an initial push to try to change the way we think about psychiatric conditions, from them being discrete disorders, you know, kind of more or less decided upon by committees, to being more continuous conditions that we might have real biologic scientific bases for. I think wisely, personally, the DSM-5 decided not to do that because although we've learned a great deal about the neurobiology of various psychiatric conditions, we do not yet have a way of unambiguously linking biologic changes to psychiatric symptoms. So, you know, psychiatry emerged, biologic psychiatry emerged in the 1960s and 70s as a way of trying to come to agreement about what psychiatric disorders were, not based on their causes, but rather based on their symptom clustering. At the time, it seemed like a very sexy idea. You know, unfortunately, the last 30 or 40 years have shown that while it's clinically useful, it really doesn't line up with psychiatric reality. So part of the challenge we face currently in psychiatry is that for clinical reasons, we still need to use these specific disorders, while we increasingly recognize from scientific perspective that that does not map on to the biologic reality. On the other hand, that biologic reality is not ready for prime time yet as a way of organizing our psychiatric conditions.